Hey there, class. This is part 2 of the video for day 5. Hey, by the way, do you like my Friday Hawaiian shirt? I thought it was kind of awesome. For this part of our video, you need to just scroll down just a little bit in the website to find the codon amino acid translation chart. That's going to become a really important piece of information for you to understand this idea of translation, where we go from the DNA, which was over there that we talked about last time, to the ribosome to eventually get a kind of protein that the cell needs. And then those proteins, in turn, make all the parts of your body. They make the hormones that go through your body. They help to make specialized cells. Proteins then do everything. So proteins are sort of where the real work happens here. With the DNA, we need the information to make the proteins so that the real work can get done. Anyway, I want to back up a little bit and remind you that in all living things, we've got 4 different types of organic molecules. Now, I don't mean the little molecules like water and carbon dioxide and oxygen, those little molecules. I mean the big molecules, the macromolecules that we talked about when we finished up our biochemistry section. And those types of molecules, you remember, were nucleic acids, and we've been talking about nucleic acids as we've been looking at how DNA works. Carbohydrates, and by carbohydrates we mean things like glucose and all the sugars that are bigger and bigger and bigger and bigger all the way up to starch, and cellulose and things like that. And then the proteins, which we're going to be talking about more today, and then the lipids or fats. So let's go back to proteins. All proteins are made up of amino acids. And I have to admit that sometimes I mix up amino acid and nucleic acid. And so if I've done that in the videos earlier, I apologize for that. It's nucleic acid that's in DNA. It's amino acids that make up proteins. So the amino acids are like the building blocks of proteins. Now, unlike DNA, they're not really a code like the nucleic acid is, but in a protein, they're literally like building blocks. So if you've ever built with Legos before, and you know how you can make all sorts of shapes with the different building blocks that go into a Lego creation? I know it's cool when you go down to the Mall of America. You see those amazing things all made out of Legos. You know, they've made Lego like full-size replicas of the Apollo capsules from the 1960s. It's just really cool. Anyway, the amino acids are like that. There's no set order that amino acids have to go in, like there isn't a cytosine always pairs with guanine sort of order. Instead, the proteins can be assembled with this wide array of amino acids, and it's the order that the amino acids go in and the way that they fit together that makes this protein what it is. It's the uniqueness of these hundreds and hundreds and hundreds of different proteins all have to do with the way that the amino acids go together. Now, if you look at that codon amino acid translation chart, you see most of the amino acids that are possible. Now, this is actually a simplified version, and sometimes you get sort of multiple codons going together to create a more complex amino acid. But this is a pretty simple way to think about how amino acids make proteins. So take a look at that chart. I've just got a paper version in front of me. You'll notice that up in the upper left-hand corner of the chart you see phenylalanine. Well, phenylalanine is an amino acid. Phenylalanine is made by the codons UUU or UUC. Okay? Now, actually, there's a little bit of difference in the way that the bonds of phenylalanine are arranged, depending on whether or not it's UUU or UUC. But for our purposes, we just need to know that both of those codons are going to make a version of phenylalanine. Okay, So that amino acid is made by those codons. So we said before that the language of DNA is that the nucleic acids, or the bases, make up words, and the codons are the words. Well, this codon then is making the word that's going to get translated. That's why this is called translation. It's going to get translated at the ribosome as phenylalanine. Okay, but there's a step in between that you need to understand. So this was messenger RNA. The messenger RNA itself doesn't make amino acids. There's another intermediate step. And that intermediate step is transfer RNA. Transfer RNA is the other side of what would be DNA if this was half of a DNA molecule. Now I've written in the uracil here to remind you that that RNA uses uracil instead of thymine, but we're still going to use pink as we demonstrate this. So transfer RNA is when you would have, for every codon, you would have a polymerase that would come along and pair up that codon with another codon. So here, here's a codon, and that's going to then pair the bases up. So remember the green guanine paired with the blue cytosine. So we've got a, there we go. And then the yellow adenine pairs with the uracil in RNA, and then the cytosine again pairing up with the guanine. Okay, so we've got a complementary codon here to this particular part. Now, I just want to point out that this is the same as this. So remember, this was the start of our process. This transfer RNA then, it gets its code and it goes to another part of the ribosome where that code, which we can read now as CAG, same as that first part of the DNA strand that we started with, that CAG is going to make now an amino acid. Okay, so go to the amino acid chart, and I want you to go down to the second row. This is where we see all the C's, the cytosine, and yours is on your computer. So C is the first letter, and then you're going to go to the third column. So this is CU's, this is CC's, this is CA's, and in the third column you find CAG, CAG, and look at what it codes for. It codes for glutamine, glutamine. So this makes the amino acid glutamine, which is abbreviated GLU. GLU. Okay. Next week, on Monday, or actually on Tuesday, because we have a short week next week, on Tuesday I'm going to give out the call to your small group, and during that time we're going to look at the mutation worksheet that's attached to the bottom of the posting for today. And one of the things that you need to do in that mutation worksheet is fill in the mRNA, transfer RNA, and then the amino acid that it turns into. So how do you get a protein? Well, you do that same process with each of these codons, and you end up with a string of protein, a string of amino acids that together make a protein. In this case, our protein string was made up of a gene that was only, if you remember, was only 3 codons long. It could be thousands of codons long to make up one protein, one gene for that particular expression of a trait that makes a particular protein. So this is the idea of translation. The DNA splits, the messenger RNA is formed by the polymerase, the messenger RNA travels out of the nucleus into the cytoplasm, where it goes and links up with a ribosome. In the ribosome, individual little bits of transfer RNA are created that now recreate the original code from the original DNA. That transfer RNA it attracts or is glommed onto little amino acids that are floating around in the cytoplasm of the cell, just ready to get transformed into proteins. And these amino acids link up together like building blocks, like Lego blocks, to make a protein. So that's all translation, otherwise known as, I'll write this word down, protein synthesis. Synthesis means to make. Protein we've talked about, so this is the making of a protein in the ribosome. Okay.